Hey everyone, welcome to The Explainer. I am absolutely thrilled you're here today. We are tackling a topic that's really as brilliantly complex as it is vital to human function, and that is bladder neurophysiology. Now, I know why you're here. You want a deep dive. My goal today is to generate a detailed explanation without skipping any point whatsoever. We are going to totally clear up your concepts, and I promise not to skip a single important detail. So you literally won't even need to go read the textbook after this. We're going completely textbook-free to give you the most structured, comprehensive anatomical roadmap out there. We're going to map the neural pathways from the very top of your brain all the way down to the sphincters and then back up again.
Okay, let's dive into this master plan. We'll start with the brain and central pathways, then move to spinal efferents and sphincters, parasympathetic voiding control, sympathetic storage control, somatic voluntary control, and finally, wrap it up with the sensory feedback loop.
All right, Section 1. Brain and central pathways. Okay, so I want you to meet the absolute conductor of our whole physiological orchestra here, the nucleus locus coeruleus. It's tucked deep down in the pons of the brainstem, and this coordinating center has one massive, crucial responsibility, and that's synchronization. See, when it's time to void, this center ensures that your bladder contracts at the exact same millisecond your urethral sphincter relaxes. Without this conductor, the bladder and the sphincter would just be constantly fighting against each other.
Now, you might be wondering, why don't we just, void the literal second our bladder gets full? Well, as adults, we have voluntary cortical control acting as the brakes. This inhibition originates specifically up in the anteromedial portion of the frontal lobes and in the genu of the corpus callosum. It actively suppresses that pontine reflex until we're socially and physically ready to go. Compare that to an infant's uninhibited bladder, right? They haven't developed this cortical inhibition yet. So the moment their bladder reaches a critical capacity, the detrusor muscle simply contracts and boom, they void.
And this brings us to a really crucial clinical anchor. If a patient gets a cortical lesion up in those frontal lobes, they effectively lose that pyramidal tract suppression. The brakes are completely gone, and the direct result of that? Urgency incontinence. Because the cortex can no longer send those inhibitory signals down to the pons, the patient physically loses the ability to suppress the micturition reflex. It's a huge clinical takeaway.
Moving right along to Section 2, spinal efferents and sphincters.
And this brilliantly illustrates the exact cross-section of the spinal cord where the magic happens. Notice those shaded blue regions, right there. The motor signals, the efferents, that are traveling down to the bladder don't just, drop down randomly. They run very specifically in the dorsal portion of the lateral columns of the spinal cord. If trauma or disease hits this exact anatomical zip code, motor control to the bladder is severely compromised.
Okay, but before we really get into the weeds with the specific nerves, we need to clearly define the two structural gates of continence down at the base of the bladder. First, you've got the internal sphincter, which operates totally under autonomic, involuntary control. You don't have to even think about keeping it closed. Then, just below that, you have the external sphincter. Now, this one is made of striated muscle, and it's entirely under your voluntary control. It's the physical gatekeeper you actively squeeze when you're trying to hold it.
Let's jump into Section 3, parasympathetic voiding control.
Let's carefully trace this specific parasympathetic route because this is the actual mechanism that makes the bladder empty. Our journey starts with the preganglionic cell bodies living way down in the sacral spinal cord, specifically in the intermediolateral gray of segments S2 to S4. From there, the fibers exit as ventral nerve roots, speeding through the pelvic splanchnic nerves, also known as the nervi erigentes. Finally, they terminate on ganglia located right inside the wall of the detrusor muscle itself, mainly in the body and the dome of the bladder. So when this entire pathway gets stimulated, the detrusor muscle contracts and the internal sphincter relaxes.
So the absolutely crucial point here is how this exact parasympathetic anatomy hooks up with real-world pharmacology. When a patient has an overactive bladder, these parasympathetic receptors in the detrusor muscle are just firing way too much. That's exactly why these receptors are the primary targets for medical treatments like anticholinergic medications or onabotulinum toxin A, commonly called Botox. By targeting these specific receptors, we can finally quiet down that parasympathetic overactivity.
Which brings us to Section 4, sympathetic storage control. All right, let's walk step by step down this completely different pathway. The sympathetic cell bodies, they sit way higher up in the intermediolateral gray column of the lumbar spinal cord, right around segments T12 through L2. The preganglionic axons rush straight through the sympathetic chain without synapsing. They just keep going to synapse at the inferior mesenteric ganglion.
From there, the postganglionic fibers branch out through the inferior hypogastric plexus to heavily innervate the bladder neck and the trigone, leading straight to the internal sphincter.
Now, at the end of that pathway, the sympathetic system basically uses two very specific receptor types to handle storage, and they work together in perfect harmony. First, you have alpha-1 adrenergic receptors. When these are stimulated, they force the bladder neck to close super tightly. Simultaneously, you've got beta-3 adrenergic receptors higher up in the bladder. When these guys are stimulated, they tell the detrusor smooth muscle to just relax. So the neck closes, the walls relax, and the bladder becomes the perfect storage vessel.
You know what's really amazing, though? It's how these systems actually talk to each other. When the pelvic nerve is stimulated, it actually causes an increase in sympathetic tone. This brilliant little interplay results in further detrusor relaxation and an even tighter bladder neck. This specific mechanism is exactly what allows a larger and larger volume of urine to be safely accommodated over time as your bladder fills, totally preventing any premature leakage.
All right, Section 5, Somatic Voluntary Control.
Remember that external sphincter we mentioned earlier, the striated muscle one? Well, here's how your brain actually controls it. The conscious signal descends in the pyramidal tract all the way down to the motor nerves in that S2 to S4 region. From there, it travels via the pudendal nerve directly to the external sphincter. This pathway is absolutely key for maintaining your continence during moments of high vesical pressure, like when you cough, sneeze, or do a Valsalva maneuver. But wonderfully, when the internal sphincter opens at the true initiation of micturition, this external sphincter reflexly relaxes to just let the urine pass.
So, we've mapped out all these descending motor signals, from the conscious brakes all the way down to the autonomic sphincters. But this begs an incredibly important question. How does the brain actually know it's time to trigger these complex motor pathways in the first place? Well, it needs information from the ground.
And that brings us to Section 6, the sensory feedback loop.
It all starts right in the bladder wall with stretch receptors. And these use two main types of sensory fibers. First, you've got the AF-eta fibers. These are myelinated, really fast transmitting fibers that give you your normal everyday sensation of bladder filling and emptying. Contrast those with the C fibers. These are unmyelinated and typically just sense noxious stimuli. But C fibers are incredibly important clinically because they are thought to be heavily involved in the involuntary detrusor overactivity we see in urogenic bladder conditions.
Let's move to and see how this builds. Let's trace this final pathway to see how it totally completes our loop. Those stretch receptors send their afferent signals up through the pelvic, pudendal, and hypogastric nerves. They enter the spinal cord at those familiar segments, T10 to L2 and S2 to S4. From there, these sensory fibers ascend primarily in the spinal thalamic tract, rushing all the way up the spinal cord to inform the brain and our good friend the nucleus locus coeruleus of exactly what is going on down there.
And there you have it, the whole shebang, a complete, super detailed physiological roadmap of bladder control, from the frontal lobes all the way to the sphincters and back again. But I want to leave you with a thought to really ponder as you digest all of this. We've seen how meticulously balanced the parasympathetic, sympathetic, and somatic pathways are, right? Given this extreme delicacy, how might a single millimeter-wide spinal lesion completely rewrite or even destroy this complex communication loop? Just think about the cascading effects on a patient's life, and suddenly, memorizing all this anatomy takes on an entirely new level of importance. Thank you so much for diving deep with me in this explainer today. Keep up the focused learning, and I'll catch you next time.